All right, go in here. Okay, so now we go into the resistance of bacterial resistance to antibiotics. Okay, so there are a few slides earlier that talks about antimicrobial, but I think I have talked enough about antimicrobial resistance and you know what they are. So over here, this is a slide that shows the latest WHO bacterial priority pathogens list. So this is to inform and guide the scientific community and also the doctors in terms of what bacteria are the most urgent in need for new drugs and what infections or bacteria that we should pay attention to. So if you want to know what are the critical, high, medium risk group, you can also listen to the recording, but very briefly, the critical groups are those that very commonly cause infection, there's high mortality available to treat the infections compared to medium group, for instance, our Staph aureus. Mortality and morbidity is moderate burden, and there are quite a number of efforts in terms of coming out new drugs to overcome this. So humans use antibiotics on bacteria. Bacteria can evolve and fight back very quickly, and that is the essence behind antimicrobial resistance. So we'll look at different ways that bacteria use to overcome antibiotics. So we briefly mentioned some of the resistant mechanism earlier. So just to bear in mind when we talk about mechanism of resistance, so this is the fight back mechanism of bacteria against antibiotics, different from the mechanism of action of antibiotics on bacteria. Okay, so I think there are 5 different. Okay, let me share this. There are 5 different major resistant mechanisms of bacteria against antibiotics. This is an image of bacteria cell and what are the mechanisms they have in place to overcome antibiotics. So look at the first one. This bacteria, they can inactivate antibiotic using enzyme. Like we mentioned about beta-lactamases, penicillinase, oh my gosh, can't pronounce, and also carbapenemase that are produced by bacteria when they encounter beta-lactam, carbapenem, for instance. Beta-lactamases can destroy penicillin G, for example, in staph or staphylococcus that we see. We can also see beta-lactamases produced in gram-negative bacteria. We also have gram-negative bacteria that are resistant to aminoglycosides by producing enzyme. Table 2 here is taken from one of the research articles that look at the important beta-lactamases in the older term Enterobacteriaceae family. So that alone, the beta-lactamases alone, there are many of them. Okay, there are a lot of studies ongoing, and the more we know, it seems like there are less hope for humans, I guess. And that's why we need to really take the antimicrobial resistance issue seriously. Second mechanism, bacteria can also reduce permeability to drugs. So if drugs target, for instance, nucleic acid synthesis or metabolic pathway or protein synthesis, if the drugs don't go into the bacteria, then that's the end of the story, right? So bacteria can reduce the permeability to drugs as a way to overcome antibiotics. For example, tetracycline, they can accumulate in susceptible bacteria. So this is a normal condition. But in the resistant bacteria, what we observe is that there are less tetracycline in there, as a way to show that there must be something that happens to the membrane. Maybe they restrict the movement of drugs into the cell as a way to overcome. And also we see that polymyxin resistance can also change. So polymyxin, remember, we say that they target the cell membrane. So the membrane structure can change and there's no binding, there's no effect. Amikacin and some other aminoglycoside resistance can be caused by an outer membrane change. So outer membrane lies outside the gram-negative bacteria. If they can't even go into the outer membrane, they then cannot go into the cell. And streptococci, for example, they have a natural permeability barriers to aminoglycoside, and aminoglycoside is not a good option to target bacterial cell. We also have antibiotic transport, and active antibiotics transport simply means that there's a approach that bacteria use to remove antibiotics out of the cell in against the concentration manner. So let's say it is water. Is that a good example? So in this case, coming back to this, in this case, active transport simply means that they either use energy in the form of ATP, or they use protein motive force to pump antibiotics from an environment that's lower in concentration against a higher concentration. For instance, outside the cell, there are higher concentration of antibiotics. So what we have observed is that bacterial cells, they can overexpress certain genes, right? For example, in this case, resistant nodulation division family genes that's found in many gram-negative bacteria. So this is the structures of the efflux pumps found in, for instance, multi-drug resistant E. coli. So we call that as a tripartite structures. So there are 3 different structures in there, lining from the inner membrane to the outer membrane of the gram-negative bacteria. So their main function is to actively pump out antibiotics from the cell cytoplasm to the outside. So if you think about what we talked about just now, the reduced permeability is one way that to prevent antibiotics from going into the cell. But if the bacteria is not able to do that, antibiotics into the cytoplasm, some of the bacteria evolve to pump these antibiotics out before they exert antibiotic effects on the bacterial cell. Or alternatively, they can alter the target or modify the target of antibiotics into the cell, or to protect the target of antibiotics so that there's no effect. So if you look at the first image here, the antibiotics, they modify the target site so that the antibiotics cannot be targeting and it's not recognized at the target site. So there's no effects of antibiotics. Or they can have a protection proteins shielding the target site away from the antibiotics. So this is bacteria overcome these antibiotics. For example, erythromycin resistant bacteria, they have a receptors on the 50S subunit of the ribosome. They change the receptors. Erythromycin cannot recognize these receptors, so there's no effect. Penicillin and cephalosporin resistant bacteria, they may have lost the PB, new PBP and penicillin binding protein, for example, penicillin binding protein 2A in Staph aureus that is not recognized, or they can increase the PBP that's present in the cell. So when there are more PBP, antibiotics have more target, so they have less effect on the bacteria. So the transpeptidation can continue in happening. The fifth one, the last one, is target bypass. The target here is the target of antibiotics and bacteria. So remember we talked about the mechanism of actions of sulfonamide and trimethoprim. They target different enzymes in the pathway of folic acid, right? So to overcome that, some bacteria, they do not, so for example, the sulfonamide resistant bacteria do not require cellular PABA anymore, and they can use a preformed folic acid. So instead of synthesizing all this folic acid, they have evolved to rely on preformed folic acid that they can take in from the environment. For in the TMPS resistant bacteria, you can see that there's additional novel alleles in the dihydrofolic acid reductase, which then increase in the target availability. So this is a bypass, and decrease the binding activities of antibiotics. and hence maintain the folic acid production. Target bypass is applicable in antibiotics that target metabolic pathway. And the target alteration, modification, and protection can be more wider range. It can be targeting protein synthesis. It can be targeting the penicillin or cell wall synthesis. So these 5 mechanisms. Let me just quickly producing enzymes that make the antibiotic useless. They can also reduce the intake of antibiotics by changing the permeability of the cell. So less drugs go in. If antibiotics gets into the cell, they can try to pump the drugs out. They modify the target so there's no binding of the antibiotics, so there's no effect of the antibiotics. So that's how they become resistant. Or they change the metabolic pathway and not relying so much on the pathways, enzymes in the process of synthesizing folic acid. So these are the 5 mechanisms of action that we see. So the origin of drug resistance is very similar to what we learned in the previous lectures that talks about the genomics of pathogenicity. So where do they get this resistance? So apart from what we have talked about before, similar to what we talked about before, they can acquire this resistance genes from the outside, either in the chromosome, integrate all these resistant genes in the chromosome, or they can get these resistant genes as plasmid. But they are also mechanisms that make them not susceptible to drugs naturally. So this is not non-genetic related. And many of the drugs, antibiotics, the drugs here refers to antibiotics. Many antibiotics target actively replicating cells that go through protein synthesis and nucleic acid synthesis. But non-replicating cells, such as cells that are older, those are in the biofilm, these drugs have no effect. If they are not replicating, they are not going through translation, these drugs has no use. And many drugs also target metabolically active microbes. If they're not synthesizing folic acid, the drug have no use. So this is non-genetic origins. Or they can lose the specific target structures. They are bacterial cells that have no cell wall. That's what we call the L form. When there's no cell wall structures, if you use penicillin, there's no effect. There's no target, right? And this microbe infect cell, the host at the site where antimicrobial are excluded. And when they are not accessible by antibiotic, there's no effect. But all these are non-genetic origin. The rest can be genetic origin, acquiring new genes and put them in the chromosome or transferred through the plasmid. So very quickly as a summary, we talk about the resistant mechanism, antibiotic resistant mechanism of bacteria. They can inactivate the antibiotics through enzyme activities. They can reduce permeability to antibiotics. They can use active transport and also efflux pump to pump antibiotics out. They can also alter the target alteration and protection, and protections of the target or target bypass. So these are the 5 mechanism of actions. Clear? This is also a summary. All good? We can move on to the third one, the in vitro antimicrobial. So this one actually we already talked about this in the practical class. So this is the method used to measure antibiotics effectiveness or susceptibility. So we can do dilution method. So this usually involve broth, less commonly agar, that has different concentration of antibiotics. And in the dilution method, we are determining the minimum inhibitory concentration. In the clinical setting, the standard concentration that is used is between 0.125 to 128 microgram per ml of antibiotics. Usually we don't test a concentration much higher than this because generally the higher the concentration, the less safe the antibiotics is to the human cell. So this is how it is tested. You have different concentration range, and those that have no growth, you then determine the lowest concentration. And that is what we call the minimum inhibitory concentration. You can also determine the minimal bactericidal concentration, MBC, by plating out tubes that have no groove and look at the lowest concentration that have no groove, reverse groove on the new agar in the absence of antibiotics. Okay, and then this diffusion method, this is a qualitative method. On one plate, you put different antibiotics and then you measure the zone of inhibition and you interpret the susceptibility of antibiotics. So this is a less tedious method, but it's, as mentioned, qualitative. And we also have the E-test or MICE test that has on the strip a gradient of antibiotic concentration. And we look for the formation of the eclipse, and the lowest zone that has no groove will be interpreted as the MIC. That's all for the lectures. That's all for the in vitro testing method. Do you have specific questions? Let me just stop share. Like very easy when I talk through the lectures. It's actually not that complicated. Do you have questions for me? Are you following? Do you have questions for me? Mechanism, actions, what else? Oh, good, sure. No questions. What about the rest of you? I checked it earlier today. I think only less than 15 people actually watched the video. So you still prefer the online lectures like this? The university is aiming for flipped classroom,